Hello and welcome to the Vortex Biosciences webinar. The webinar is entitled the VTX-1 Liquid Biopsy System for the Flexible Isolation of Intact CTCs, an example of TetherChip microtentacle analysis. It's an absolute pleasure to be your moderator today. My name is Chris Howey and I'm Head of Sales at Vortex Biosciences. And this is the first of what we hope will be a series of webinars. We have delegates from all over the world, so I'm keen to facilitate an engaging and informative session with our excellent speakers today. By way of introduction, we have Professor Stuart Martin, University of Maryland School of Medicine. We have Dr. Elodie Solier, co-founder at Vortex Biosciences and scientific advisor. And we have Dr. Adrian Kincaid, new CEO of Vortex Biosciences. So in terms of agenda, it's going to be very interactive, hopefully 90 minutes. We have a quick introduction from our new CEO, Dr. Adrian Kincaid of Vortex Biosciences. Then we will pass the baton over to Dr. Elodie Solier to kick off the webinar for 25 minutes. And then Professor Stuart Martin will then talk for 40 minutes. We're hoping for a very interactive question and answer session for the last 20 minutes. And the webinar will finish at 5:30 UK time. So just some housekeeping before we begin. As I mentioned, we're looking for that interactive Q&A session. So please use the Q&A tab at the bottom of the screen as the chat facility has been disabled. And thank you indeed for joining. We really appreciate it. And should you have any further questions after the webinar, please feel free to contact me by email. So without any further ado, I'm going to stop this sharing and pass the webinar Zoom baton over to Adrian to say some introductory words. Thank you very much.
Thank you, Chris. My name's Adrian Kincaid. As Chris said, I'm the incoming CEO of Vortex Biosciences. It's great to be online today, and especially with such an interesting webinar to look forward to. I'm delighted to have joined the Vortex team, and it's a great opportunity to play a part in the next phase of development of such an interesting business. For me, Vortex is an enviable position with an outstanding core technology and a class-leading platform to provide viable CTCs with remarkable efficiency and purity. More importantly, I think the VTX-1 is an enabling technology platform which is able to furnish CTCs for groundbreaking science, such as we're about to hear about today. Thank you very much, everyone, for joining us today, and I'll hand you back to Chris. Thanks.
Thank you, Adrian, for those kind words. Just by way of introduction, before Elodie takes the stage, I just want to introduce Elodie. Elodie Solier received her PhD from Grenoble Institute of Technology and a CEA in France, which she completed her postdoctoral research studies at UCLA under the mentorship of Dr. Dino Di Carlo. Elodie then co-founded Vortex Biosciences and acted as Chief Scientific Officer. Elodie is now located in France, where she supports Vortex Biosciences as a scientific consultant. Thank you, Elodie. Over to you.
Thank you, Chris. Thanks for the introduction. And can you see my screen? All good. So the webinar today, the first of a series, will present the VTX-1 Liquid Biopsy System, a technology that is enabling the isolation of intact circulating tumor cells from blood samples. That's a technology that has been developed and is commercialized by Vortex Biosciences, which is a spin-off from UCLA, created in 2012 and now located in Pleasanton in California. So at first, a quick reminder or introduction for the people in the audience that may not know yet what CTCs are. So CTCs, or circulating tumor cells, are cancer cells that detach from a primary tumor, enter in the bloodstream, propagate through the body, where they're gonna stop on other organs of the body to create secondary tumors. These secondary tumors are also called metastases, and they represent 90% of the cancer-related death. Even more, there is now some belief that these CTCs may actually be present earlier before the primary tumor, before that can be detected by conventional imaging techniques. That's, for example, the conclusion of a study from Professor Hoffman in Nice with heavy smokers population, where they could find CTCs before the detection of the tumor by a CT scan. So that's why there is really this interest, this growing interest for these seeds of the metastasis, for these CTCs. So this interest for isolation of the CTCs, detection, enumeration, and analysis. But the isolation of these cells is very challenging because they are very rare and surrounded by millions of blood cells, such as white blood cells and red blood cells. So that's really challenging to get these cells, and people always use the metaphor of a needle in a haystack, while actually you just put fire to the haystack and you get the needle. Here it's quite more complicated than that. But still, yeah, there is this interest for the CTCs because they provide a real-time window of the cancer of the patient all along the cancer care, so all along the cancer evolution for each patient. And that's especially interesting when the tissue biopsy itself is not available anymore, or not easily accessible, which is quite often the case for specific cancers, such as lung cancer, for example. So there is this growing interest for the CTCs, as I said. That's why several technologies have been developed over the past years to isolate the CTCs. There are either approaches that are going to use the markers expressed by the cells to catch them and pull them out of the blood flow, or some small features like filters, where you're going to push the blood and use the size aspect to keep the larger one, which are the CTCs, while the smaller elements, for example, the blood cells, may go through. So there are different types of approaches, but still, Cell Search, that you can see here from Johnson & Johnson and recently acquired by Deparey, remains still the only FDA-approved technology for CTC isolation, specifically for their enumeration as a prognostic marker of colon, prostate, and breast cancer. So that's the only still FDA-approved system, and that's composed of two machines. So you have a machine that is going to get the cells with the epithelial markers they express. And then another machine where you bring on the cassette with the cells, with the CTCs in, that is going to image and count the cells. But over the past years, the market demands have significantly evolved for two reasons. First, because now there is this common understanding by all actors of this of the cancer community that there is way more clinical value to the CTCs than just their enumeration and just their prognostic value. So there is way more than that. And also the fact that some CTCs may not express the epithelial markers that are needed to enable such label-based approach for the capture. So by using this kind of technology, you may miss some of the CTCs that don't express the relevant markers. So that's why there is a need for more than CTC enumeration, for a free access to the cells, and also a need for a label-free capture. This is why we have developed this technology from 2012, the VTX-1 Liquid Biopsy System, with a clinical mindset, with a clinical vision moving forward. That's why we developed it in a fully automated manner. So what does that mean? So you have two parts. First of all, you have a single-use plastic consumable here. So the user take a tube of blood, connect the tube of blood to the cartridge where the blood is going to be diluted by the machine. The user also selects a container of choice to collect the cells, the CTCs in. That can be for Petri dish, Eppendorf tube, glass slide, and so on. Then the user brings that into the machine you see in the middle here, and then press go with the user interface and comes back to get the cells out. There is no sample preparation. There is no need to babysit the machine. The user can do whatever experiments they want in parallel, like on the bench or so on, or prepare even. They can even prepare the next steps of the process. So this technology has been designed obviously with the performance in mind. So to guarantee the best recovery of the CTCs, so to get as many CTCs as possible, but also keeping in mind the contamination, which means as less white blood cells as possible from the output to enable specific assays downstream. The process is purely fluidics, and I will show you how it works afterwards. So we don't use any fixatives. The cells are not physically constrained in the system or in the consumable, which means that they remain intact, viable. Process is very simple. So for the user, at least, so the user puts the blood in and gets the cells out. And label free, so no antibodies, no tagging of the cells. They are like free in suspension. And also flexibility for the input and for the output. What does that mean? So flexibility in the volume of blood, because we know that depending on the countries or the hospitals, the requirements in terms of blood collection by the ethical committees may differ from a site to the other. So the volume of blood available is not always the same. So here you can process whatever blood volume you have. And also flexibility in terms of collection, so output, in the sense that the user decides what assay they want to do. So they select the container of choice, and the machine is going to drop a given volume of cell output directly in the container. So there is flexibility for the downstream assay. All of this has been published, and I will refer to our publications all along the presentation. But if you want to know more about the product itself and its performance, that's one of our publications that is available on our website. So that's the system as it is commercialized now. How does that work? So we use inertial microfluidics. So you have here on the left side a picture of the consumable of this plastic chip with like the blood coming in here and flowing through the channels. And here you have a zoom. So we inject the blood. So no lysis is needed, no mixing with reagents and so on, just the blood and the diluent buffer. So the blood is injected at a high flow rate that is going to generate lift forces that are going to push the cells away from the walls and from the center on some kind of equilibrium positions. And as the blood is entering these openings, these reservoirs, where there are some whirlpools, the cancer cells are going to get trapped in these whirlpools while the blood cells keep flowing. So all of this is, again, quite explained and demonstrated with cell line spiking experiments in various publications over the years. The key aspect here is that over the years, this design has been optimized with more reservoirs in parallel and series to really enable a high throughput of the blood processing. But I think the best way to show you how it works is to show you a movie. So this is a high-speed movie of what's happening really in the chip. So these are breast cancer cells, MCF7, spiked in healthy blood. So you saw the cancer cells getting trapped in these vortices. Now they are getting washed from all the blood contamination. And when this is washed, we stop the flow, which is going to dissipate the vortices and release all the cells of the chip. So again, the white, larger one are the cancer cells. They are getting trapped over time. the blood cells are flowing away with the wash step that is typically done with PBS, but this liquid can be adapted. We stop the flow, vortices are dissipated, and the cancer cells are leaving the reservoirs to be directly collected of the consumable into the container of choice. As you can see, this is continuous flow in the sense that there is no retention of the cells in the very small features. So the cells are always flowing and they are in suspension and freely and happy. So obviously with a patient sample, you may have less cells than that. So here it was really a spiking experiment. So the workflow, as I said before, we take a tube of blood, we connect the tube to the cartridge, we bring the cartridge into the machine. We have validated processing from 200 microliters for mice blood up to 16 cc for patient samples. You come back after a given time and then you get the cells out. This time here depends on the mode that you select on the machine, in the sense that the user can select either high purity or high recovery mode. What does that mean? When the blood is injected through the chip, we can take the waste and re-inject again and again and again to maximize the recovery of the CTCs. But that means that each time, each cycle, you get more cells, but you also get more white blood cells as well. That's why it's a compromise between the recovery of the CTCs and the purity you need, which is why we call that high recovery mode, so 3 cycles, or high purity mode, less CTCs, less cycles, but also less white blood cells. So the number of cycles you do will trigger, obviously, the timing of the overall experiment. Then we, at Vortex and the R&D team, and through collaborations with KOLs, we have validated different workflows. And by validating, it means that we have optimized the workflows internally and with these collaborations to guarantee the best recovery and to guarantee the best assay downstream. And we always end up such optimization work through application notes or application protocols that are shared with the customers, or ideally with also peer-reviewed publications. So you have example here at the bottom of some publications that were done with some users to really share the optimized protocols we recommend. Obviously, at the end of the day, the users are the experts and they decide what they want to do with the cells. We are just here to guide them through the process. So among these workflows, I can cite genomic analysis. So for example, for DNA, some users have done Sanger sequencing, next-gen sequencing, qPCR, targeted qPCR, for example, for EGFR. On RNA, there has been some RT-qPCR assays done on the CTCs, pooled CTCs collected, or also combination with 10x genomic platform for single CTC RNA sequencing. Some users have done live cell assays, for example, clonogenic assays, or for example, tentacles analysis, as you will learn later. Obviously, cell staining, so immunofluorescent staining, and also enumeration, but also cytology, FISH, so more conventional technologies, more conventional assays, and proteomics. So a user had their own Western blot platform that they brought after Vortex, so to collect the CTCs directly in their chip, and they did single CTC Western blotting from breast cancer patients. So again, all of these protocols that have been optimized, where we have tested different commercial kits and so on, all of this is publicly shared through application protocols or scientific publications, and all of this content is online available for the users. Data recovery. So whenever we start a new cancer type, we always recommend some cell spiking experiments. So spiking of a given number of cell lines in healthy blood. Then you know how many you brought in and you know how many you recover. And you can, this way, you can estimate the recovery. So 60–70% is the range we got at Vortex for MCF7 breast cancer cell lines. This recovery varies depending on the cell lines, obviously. 1 hour per sample, this is for one cycle processing of a sample. But as early as possible, we strongly recommend to switch to real patient samples because cell lines are a good model, but this has some limits, and it's really important depending on the assay you want to do downstream. So these data here are CTC numbers. So the y-axis is the number of CTC per tube of blood, so 7.5 cc's EDTA tube, for patients with metastatic cancer, breast, lung, and colorectal cancer, but going under therapy. So some of these patients are going well and some others not as well. And we counted here the CTC number using immunofluorescence-based staining and counting, manual counting. So in this case, either CK, EpCAM, or cytokeratin and EpCAM for the 3 studies. So that's the number of patient samples. So 1 dot being 1 patient sample. And that gives you a range of CTC number for this specific study. We always, with new instruments, and we do that very often, we have this quality control internally to test for the performance, the reliability and repeatability from a run to the other, from a unit to the other, from a user to the other as well. Where we test for the recovery of the cells. So in this case, it's MCF7 spiking experiments. So recovery and purity. So in this case, contamination, because we are looking at the number of white blood cells per milliliter of blood process. So that gives you an idea of what is our passing criteria, 60% for the recovery and less than 100 white blood cells per milliliter, and a standard deviation less than 10%. So here these were 4 runs of 3 cycles from the same tube of blood, so different aliquots of the same sample for each unit. So that's an indication of the reliability, the repeatability of our instrument. And that gives you an idea of the criteria we are looking for to assess whether an experiment is okay or not. Again, that's different than a real patient sample. These are with cell line experiments. So this one is represents some CTCs extracted from colorectal cancer patients. They show here that we have different staining, so different patterns. So because our capture is label free, we can capture epithelial cells here in green, so EpCAM CK positive, but also mesenchymal cells in orange, vimentin and cadherin positive. So either cells that are mesenchymal or epithelial, but also both, like at the transition stage where they express both types of markers. So that really shows we can capture a broad range of CTCs in terms of expression, which would not have been possible with a label-based approach, but also that we can have single cells and clusters. You can see here a cluster of mesenchymal CTCs. That can be of interest because some researchers in the community believe that these mesenchymal clusters may actually be more aggressive than single cells. I mentioned that briefly, but we have also developed a workflow for mice blood sample for cardiac puncture, where we can go as low as 200 microliters of blood that is diluted then with PBS and brought into the VTX-1. We have also set up an immunofluorescence staining protocol to stain for the mice CTCs or human CTCs into mice. So this was then applied by a user, a KOL at Stanford, Vishnu Ramani and et al., where the breast tumors were implanted into mice. So these were patient-derived xenograph. And this graph here shows you the number of CTCs per 100 microliters of cardiac blood over time from the implantation days. You can see here that over time, the number of CTCs is drastically increasing along with the tumor volume and then the appearance of the metastasis in the mice. So we have a nice correlation between the number of CTCs and the spread of the cancer. Also some pictures of CTCs, either single or clustered. And interestingly here, there were some homogeneous clusters or heterogeneous clusters. What does that mean? It means a mix of CTCs with also other cells, so white blood cells here that were CD45 positive. So this is published as well if you are interested to learn more about it. An important point, an important differentiator for our approach is the fact that we collect the cells intact. So that enables the users, such as Professor Martin, to have direct access to the cellular entity to study cancer biology and many cool things. So we always test that with A549 cells, so cell lines spiked in blood. We confirm here that they proliferate after the processing, but also that they keep their capabilities, their functions. So in this case, a user demonstrated that they can still form colonies after vortex processing, or also that they keep their invasion capabilities, so to form here spheroid structures and invade an extracellular matrix. And so some users can also look at the tentacles and how they form and how their formation is impacted by the use of specific drugs. But I don't want to say too much about it. Professor Martin will describe much more that in a very short moment. Wanted to point out an important aspect of this of the CTC processing and the way the VTX-1 is used and could be used in the future. And that's also a topic of interest for all liquid biopsies in general. That's the crucial importance of the blood collection tubes. That's important to consider as we want to bring VTX-1 to the clinic, because you may need to ship blood from a site to the other. And also the blood may have to wait all the samples for a batch processing, for example. So all of this, if we want to move to the clinic, that has to be considered. That's the first point. And the first point is that depending on the assay that the users want to do downstream, the tube that is used is very crucial as well. If you want to do enumeration on a glass slide or RNA assays or live cell assays, the anticoagulant won't be the same. The blood collection tube won't be the same. So in this case here, we have, for example, compared different blood collection tubes that are commercially available. EDTA is always our reference tube, so the purple cap. And we always compare these tubes with MCF7 cells again, spiked in healthy blood. And day 0, day 1, so 24 hours and 72 hours here. So just be aware that this data exists and that the recovery is conserved for EDTA cell save, a bit lower recovery, but still a good number of CTCs with Treg tubes and with LB guard. Then after 24 hours, the recovery is expected and is indeed dropping a bit, which is then why the blood collection tube has to be carefully considered if a shipping of the blood sample is needed. The storage temperature as well can help depending on what assay you want to do downstream. So in this case, the partner really wanted to preserve the CTC's viability and the transcriptomic content and wanted to use citrate A. So we confirmed that recovery was similar between citrate and EDTA, either at 4 degrees and room temperature. And that after 24 hours, that was still acceptable with a decrease at 48 hours. So this was really designed in view of transcriptomic assay. And again, if you want to do enumeration, fixation enumeration or live cell assays, this has to be considered if it's above 24 hours. Less than 24 hours, EDTA is perfectly fine. So I shared some of the data, but we have much more, and genomic assays, for example, so many more studies. All of these studies have been published, and the publications are available on our website if you want more information. As much as possible, even if it has been difficult this year, but we always try to present at conferences our progress, our research by users. So again, this is a lot of it is shared on the Vortex website. So feel free to go, and in any case, if you need any, if you have any questions, you can use this email address or the email address from Chris directly, chris@vortexbiosciences.com, or mine, elodie@vortexbiosciences.com. And thank you for joining us today. And Stuart, all yours.
Thank you, Elodie. Before we dive in, I really appreciate that fabulous presentation. Really excellent. So thank you. I've seen the questions coming in. So that is excellent. So keep those coming in. Stuart, before I hand the baton over to you, if I could just introduce you, that would be fantastic. So sure. Professor Martin received his PhD from the University of California in San Diego. He completed a Damon Runyon postdoctoral fellowship on breast cancer metastases at Harvard Medical School under the mentorship of Dr. Phil Leder. Dr. Martin is currently a professor at the Greenbaum Comprehensive Cancer Center at the University of Maryland School of Medicine. And just your focus, Stuart, if that's okay. So Dr. Martin's lab studies mechanical properties of circulating tumor cells by combining approaches from physical sciences and engineering. His lab discovered the novel microtentacles on the surface of CTCs that promote metastases that he will tell you a bit more about today. So thank you very much, and I pass the baton over to you. Thank you, Stuart.
Okay, great. Thanks very much, Chris. It's really a pleasure to be here and to tell you about how we're using the unique Vortex technology in our studies. So hopefully you're able to see the screen there, as well as my pointer. And so Elodie mentioned that we study these structures called microtentacles in my lab, and the unique capabilities of the Vortex have really helped us to capture viable CTCs for these studies. And so just starting off, my lab focuses on breast cancer, but of course, it is not the primary tumor in breast cancer that really threatens the life of the patient. It's once those cells spread to distant sites through metastasis. Now, the challenge we have in the clinical treatment of breast cancer and many other cancers is the detection threshold. So by the time a metastatic tumor appears on an MRI or a PET-CT, it's reached a size of a focus of more than 10 million tumor cells. So we're really seeing metastatic recurrence at a very late stage. Now, it's been appreciated, and part of the impetus for the development of isolation of circulating tumor cells is we know that even small primary tumors will shed circulating tumor cells into the bloodstream at a very early stage. Since those tumor cells are essentially too large to fit through capillaries, they get stuck in the first capillary bed they encounter. Some of these cells are pushed through by blood pressure and fragmented. Others, a small percentage, will escape the blood vessel and persist dormantly in the distant tissue. Now, the tumor cells go dormant because the environment of the metastatic tissue is different from their original environment. So these cells shut down and stop dividing, which of course makes them almost entirely resistant to traditional chemotherapies that target dividing cells. So these stages of metastasis of seeding and dormancy exist below our clinical detection threshold via traditional clinical imaging methods. And we also don't have good drugs to treat these stages of cancer metastasis. Now, in the lab, we can use mouse models, and we specifically express firefly luciferase in the tumor cells that are injected into the mice. so that now within minutes of when the luminescent tumor cells are injected into the bloodstream of this mouse, you can see they trap within the capillaries of the lungs of that mouse. Then about 90% of the CTCs are lost over the first few days. And then in the mouse models, within a few weeks or months, they recur, in this case, within the lung. So our lab, like many other labs, has been interested in how do we eradicate these tumor cells at an earlier stage to delay or eliminate metastatic recurrence. And when I first moved my lab here from after Harvard Medical School to the University of Maryland, we were interested in focusing on the specific mechanisms of circulating tumor cell metastasis. And this grew out of an observation that we made when we took breast tumor cells and put them into a free-floating environment where the tumor cells couldn't attach to mimic what was going on in either the bloodstream or lymphatics, the cells free-floating. And when we looked at the breast tumors in these free-floating environments, what we saw was they generated these long extensions of their surface that we'd never really seen before. My work at Harvard had been on the actin cytoskeleton, so we initially assumed that these were actin-based filopodia. So we did an initial test where we took these free-floating cells with the short projections. We treated with a drug that inhibits actin polymerization that was known to destroy filopodia and invatopodia on tumor cells. And we made the prediction that if these structures were based on actin, they would go away when we inhibited actin polymerization. Well, they didn't go away. They got longer and much more numerous, which showed us they were not based on actin, and the data actually suggests that they're suppressed by actin. When we followed up with immunofluorescence, what we found were there were microtubules along the lengths of these structures, and actin was really only found at these sort of dense ends. And so they were a structure that hadn't been reported before. We named them microtentacles. And the reason we named them microtentacles is they help the tumor cells aggregate together. So that if we look at a confocal microscope image of two free-floating breast tumor cells that are labeled on their surface with the red plasma membrane dye, we can see they're forming tentacles. We can see the cells are attaching to each other, but we can't tell where one cell ends and the other begins. If we label the cell on the left with a membrane localized green fluorescent protein and then look at what we can see, what you can see in this case is the tentacles extend around the neighboring cell and help hold the cluster together. And of course, there's tremendous interest in the clustering of circulating tumor cells at this point, because circulating tumor cells that form clusters have been shown to have up to 50 times higher metastatic potential. And so we're currently targeting microtentacles as a therapeutic intervention to break these clusters apart. The other thing we were able to show through this same sort of live cell confocal imaging was if we took a green labeled tumor cell and allowed it to attach to red labeled endothelial cells that would normally make up the blood vessel wall. Again, we reconstruct a three-dimensional image of these cells together. And what we can see is the tentacles are penetrating through the endothelial cells. And when we cut through in cross-section, we could actually see that the tentacles are actually penetrating the junctions between the endothelial cells to help the circulating tumor cell reattach. So over the years, we've developed a number of different therapies that could potentially be directed against tentacles. This one, tetracaine, is a kinesin inhibitor that makes the tentacles collapse by targeting the vimentin intermediate filaments that are within the tentacles. So this is a time lapse of about a 10-minute drug effect. And so what you can see here is the tentacles are moving rapidly, and as the tetracaine takes effect, the tentacles pull back into the center of the cell. So we do have a number of different therapeutic candidates for targeting the microtentacles. The other thing we've learned about this is since the tentacles are based on microtubules, there are many existing cancer therapies that already target microtubules to either destabilize the microtubules or stabilize them. And so in the case of a tubulin stabilizing drug like paclitaxel, if you take a tumor cell that has very few tentacles and inhibit its growth by stabilizing microtubules, since the tentacles are based on microtubules, the cell treated with paclitaxel gets more tentacles and reattaches faster. So this raises the prospect that some of the drugs that we're using in the clinic right now, even though they reduce tumor growth, they could be increasing the metastasis of circulating tumor cells. And this is something we've been discussing for years, but there have been emerging studies from other independent groups like this one at Albert Einstein College of Medicine, Maya Oktay's group, together with John Condoleezza and George Karagiannis, reported in Science Translational Medicine in 2017 that if they gave neoadjuvant chemotherapy before surgery in mouse models, this caused the following effects. Macrophages were recruited to the tumor. You got bursting of the tumor blood vessels, shedding of circulating tumor cells into the bloodstream, and it actually increased metastasis in 3 different mouse models. In addition, similar molecular changes were seen in patient tumor samples after the application of chemotherapy. And importantly for us, with respect to tentacles, this occurred with paclitaxel treatment. But interestingly, it also occurred with other types of chemotherapy, like DNA damaging chemotherapy. So this may be a much more common effect to scatter circulating tumor cells through the use of chemotherapy than has previously been appreciated. So we became very interested in testing this in live patient tumor cells. And so what we showed was if we took patient tumor cells at the time of surgery and immediately put them under the microscope, so these cells had only been out of the patient for a few hours, what we can see is those cells are already forming tentacles within these first few hours. If we treat with a microtubule destabilizing therapy like colchicine, this is a 30-minute drug response in the same cell. What we could see is collapse of the tentacles in only 30 minutes. So this gives us a platform if we can get live tumor cells to actually test patient drug responses in less than an hour. The challenge that we had was how could we streamline this and how could we apply it to circulating tumor cells? And so part of the issue was how do we image the free floating cells, because these tentacles only occur when the cells are free floating, like they would be in the bloodstream. And so we had seen also through collaborations with the University of Patras that microtentacles help hold together. So these are from patient CTCs. Microtentacles help hold together clusters of CTCs and also clusters of CTCs together with white blood cells. As Elodie had mentioned, the CTCs can cluster with themselves, or they can actually cluster with the cells of the immune system. And so we were interested in developing technology to streamline this, both for collaborators and also for clinical studies that we have ongoing here. And so the challenge that we had was it's very difficult to image a free-floating tumor cell. So we use confocal microscopy, where you can image a single plane of the cell. And since tentacles exist in 3 dimensions, we see them going in and out of the plane of focus. And of course, if we take a series of images to reconstruct a 3-dimensional image, the cell drifts and blurs on the microscope. So we needed to solve this problem. And the way we did it was through collaboration with a group of Dr. Christopher Jewell in the bioengineering department at the University of Maryland. And together with Dr. Jewell, we developed a nanosurface for microfluidics that we've named the TetherChip. And basically, this is nanometers thin and optically clear and prevents the cells from attaching. And then into the top of this surface, we incorporated a hydrophobic lipid that binds with the cell membrane. And this allows us to image microtentacles. It allows us to follow things like stem cell spheres, as well as clustering assays and deformability of the cells. And so the overall advantages is this is an optically clear, very thin surface. It spatially immobilizes the cell. It maintains their free-floating characteristics, and we can test drug responses in less than an hour. And it's also a fairly simple to manufacture technology. And so looking now at some cells on the TetherChip surface, what we're looking at here is 5 optical slices of the cell so that we can see the tentacles. But if we look at them as in real time, we can see the tentacles moving. If you look carefully in the background, you can see debris flowing past, and that would normally wash the cell out of the field of view. But in this case, the cell stays tethered right in the center, but continues to do what it would do when it's free floating, form these tentacles. If we now wash in a drug that destroys tentacles like colchicine, we can see them collapse. Or if we use a drug like Taxol that actually stabilizes microtubules, we can see they get longer, more numerous, but also a little bit more restricted in their motion. So through this capture technology, we were also able to refine our image analysis so that we could actually see if we traced the perimeter of the cell and then wrote a custom program that was reported in this publication here that found the points of local maximum curvature in this perimeter line that identified the ends of the microtentacles. And so now even in the live samples, we're able to trace the microtentacle metrics in terms of number per cell, average microtentacle length, and those sorts of things. So working both in the development of the device and also in the development of the analysis software really prepared us to undergo clinical studies. But now what we needed to be able to do was isolate live circulating tumor cells in order to do that. The other advantage we had through the TetherChip technology, and something that we had appreciated during our years of studying these structures, is microtentacles are exceptionally thin and they're very sensitive to chemical fixation. So if there's a fixation step involved in the preparation of the sample, it destroys the tentacles. And so we were able to demonstrate this on the TetherChip. So this is a cell that's been attached to the TetherChip, but its tentacles are still freely moving. They haven't bound down to the surface. When we add a fixative like formaldehyde that's used in the preparation of many clinical samples, you'll see a brief flash in this movie when the formaldehyde gets added, and it'll be obvious what happens to the tentacles. So you can see they're moving slightly. When the formaldehyde gets added, the tentacles deflect, and they almost immediately begin to collapse. If we instead allow those tentacles over the process of about 30 minutes to bind to the surface of the TetherChip, we've engineered the TetherChip so that it remains reactive to formaldehyde, so it's able to preserve the tentacles. So these tentacles have now attached down to the surface of the TetherChip. Again, you'll see a brief flash when the formaldehyde comes in. And I think if you look very carefully, you might see this one tentacle here bend a little bit. But go here, you'll see a flash again when the formaldehyde comes in. And basically the tentacles are preserved. Again, you do see this one here bend a little bit, but overall they're preserved. So this now gives us a streamlined clinical workflow to actually go rapidly from the isolation of live cells to testing their drug responses to preserving that through chemical fixation. And so in this publication here that we did also together with using the Vortex technology, we were also able to extend this to look at microtentacles in colon cancer cell lines, lung cancer cell lines, prostate cancer cell lines, and we see tentacles across this wide variety of cancer cell types. And so then the great advantage to us of the Vortex technology is it's this rapid non-contact isolation of live circulating tumor cells that Elodie told you about. And importantly, as Elodie mentioned, you can integrate your own capture vessel into the VTX-1 instrument. So this enabled us to get a seamless integration with the TetherChip, because basically the tumor cells that come off of the Vortex chip, in this case within the VTX-1 instrument, they go directly onto our TetherChip. And for these captures from blood samples, we actually use a long single channel TetherChip that has a volume of about 300 microliters to capture all the cells that are eluted from the Vortex chip. And so because we don't have to subject the cells to a lot of physical pressures, because we isolate them very quickly, we're able to capture the tentacles very well with the combined VTX-1 and the TetherChip. And it gives us very nice images of tentacles that then captured on the TetherChip and subjected to chemical fixation, we get preservation of the tentacles. And so these are just two examples here. You can also see in this one image here that we had a few contaminating red blood cells, but I think you can appreciate the difference in size between the red blood cells and the tumor cells that are generating tentacles in this case. And so that's the principle on which the VTX-1 is isolating the cells based on their size. So it's label-free and it captures the live cells. And that's been an incredible advantage to our studies. And so carrying this forward, what we did was we had breast tumor cells that were spiked into the blood samples that we isolated on the Vortex. And then we treated for 1 hour with a microtubule destabilizing therapy, vinorelbine, which is FDA approved. And then we could subject those to the automated image analysis to look at the length and number of microtentacles on the cell and the average microtentacle length, either before vinorelbine or after vinorelbine. And so then these are the individual cells analyzed for that drug response. And the differences that we get between these populations are quite significant, to the point where it shows us that we could generate 90% power with as few as 11 tumor cells, which is therefore reaching feasibility for both patient biopsies and CTCs, where you do get very few cells. And so this allows us rapidly within 1 hour to test those live cells for that drug response. And so we've recently translated this to preclinical models. We prioritized vinorelbine because of its ability to reduce the microtentacles, but we wanted to test whether vinorelbine would have a selective effect on the metastasis of the circulating tumor cells. So we needed to devise a way to test that principle in mice. And so the way that we did that was we only treated with vinorelbine for a short pulse of 24 hours. When the tumor cells were injected into the mammary gland, that 24-hour drug treatment didn't have a significant effect on the growth of the tumors and didn't affect the survival from the breast tumors, because again, they only got 24 hours of drug. If we gave that same 24 hours of drug at the time that the cells were injected into the bloodstream, under the vehicle control conditions, the cells trap in the lung, they're lost over about the first 24 to 48 hours, but then they begin to recur by about 3 weeks, and many of the mice succumb by 7 to 8 weeks. If we do the same 24-hour treatment with vinorelbine at the time that the cells are in the circulation, we see that there's reduced seeding of the cells within the lungs, but importantly, the recurrence is strongly delayed, so that the vehicle control mice die relatively quickly, and that single 24-hour treatment with vinorelbine extends survival significantly. In fact, the median metastatic tumor survival improved from 8 weeks to 30 weeks with just that 24-hour drug treatment. And so this was showing us that the microtentacle analysis in live tumor cells can rapidly identify drugs capable of reducing CTC metastasis. And importantly, an identical treatment could possibly be completely overlooked by tests that are dependent on tumor growth, because this 24-hour drug treatment with vinorelbine didn't affect primary tumor growth, but it does significantly improve metastatic survival. And so this is really the opportunity now to use technologies like the VTX-1 to enable this sort of precision medicine strategy for patients in the clinic. And so this is just one example here of the microtubule-based therapies. The current ones that are FDA approved either destabilize microtubules or hyperstabilize microtubules, with the common goal of inducing mitotic arrest, tumor cell death, and delayed recurrence. The problem with this is that we've just seen from the example of paclitaxel, which is a taxane, stabilizing microtubules is also associated with a number of phenotypes that would not be desirable for the cancer patient, in that it imposes wound healing effects, upregulates modifications of microtubules like acetylation and detyrosynation, and microtubule stabilization through tau that are associated with poor prognosis, increased migration and invasion, EMT, microtentacles, stem cell characteristics, et cetera. So existing chemotherapies that we have, there may be a dramatic difference, even though they both have similar effects on tumor growth, into how they promote metastatic outcomes. And so our overall goal here is now to implement this for patient primary tumors. One of the gold standards, at least in the area of breast cancer research, is to take freshly isolated patient cells, transplant them into mice as patient-derived xenografts, and then grow out those cells for further studies. The advantages of this is it retains the original molecular markers of the original patient's tumor. It's an indicator of patient prognosis if the tumor cells grow. And interestingly, it also mimics the metastatic pattern of the original patient. Now, those are the advantages. The disadvantages are PDX growth and metastasis often takes longer than 6 months. And so to grow out those cells and then transplant them in different mice to conduct a drug study could easily take you a year or more. In addition, more than 50% of the tumors from patients never grow as PDX in mice. So you lose the representation of those patients with a good prognosis signature. So we've decided to take a different approach. While many groups are developing more complex methods to actually grow cells out, we've decided to focus on what can we analyze in the patient cells immediately after their isolation. And using the TetherChip, we can look at microtentacles, which I've told you about. We can also look at the ability of the cells to cluster or their ability to form stem cell spheres, and decide which of these phenotypes are now indicative of tumor genesis, metastasis, and drug response. And so importantly here with the sphere formation, that's an assay that usually takes about 7 to 14 days. We've shown with the TetherChip that we can actually shorten this also to less than 24 hours, which is what we can do for the others. And the reason for that is the TetherChip actually holds the cells in place. And so traditional sphere forming assays are done in a free floating low attach condition. When we compared that with tethering, for small numbers of cells, like 10, 100, or 1,000 cells, what we found was once we got to 1,000 cells, the cells on the tethers were forming spheres more efficiently. And of course, these sphere assays are traditionally done with more like 10,000 or 100,000 floating cells. And what we discovered from this is, of course, the traditional sphere assays are combining the phenotype of a single cell growing into a sphere and the ability of the cells to cluster together because they're all free floating. So if we mix together 500 green cells and 500 red cells in a free floating environment, we could see under the traditional sphere forming assays, we got multicolored spheres because the cells clustered together. On tethers, our spheres grew out as increasingly as a single color, showing that the cells are growing in place as single cells. And so this is also shown here. If we trace the same field of cells from the first day to day 5, we can see these initial cells here then grow out as spheres. And so this allows us, if we can get live circulating tumor cells, to actually measure what % of those cells will actually successfully grow into spheres. And what we showed here is if we looked after 24 hours into the cells that went from a single cell to 2, 3, or 4 cells, we could predict with 97% accuracy which were going to form stem cell spheres by 7 or 14 days. So this is how it allows us to speed up the assay. All right, and then finally here, I just wanted to show you some imaging that we're currently doing in zebrafish as we try to identify which FDA-approved therapies could actually reduce the efficiency of circulating tumor cells. We're imaging a zebrafish here that has its blood vessels labeled with GFP, and then injecting into the yolk sac fluorescently labeled tumor cells. And when we image this with a light sheet microscope that can go 30 frames per second, this is what you see. So here's the zebrafish head. We can see its heart beating here with this flutter. And this is now the vena cava leading up to the heart. And you can see the injected tumor cells here are actually being pulled into the heart. If we look with a little bit higher magnification, we can actually see a whole parade of the injected tumor cells being pulled into the heart and projected out through the arteries into the veins of the tail. So this was with the light sheet microscope where we could go 30 frames per second. But we used a spinning disc microscope to image the cells in the veins of the tail, which could go 200 frames per second. And then we can see incredible detail of the tumor cells actually rolling along the blood vessel wall and actually using microtentacle extensions to reach out here for the blood vessel wall. So this now gives us an in vivo model where we can trace the mechanisms by which circulating tumor cells reattach to the blood vessel wall. All right. And so we're translating this, our first step to translating this into both retrospective and prospective clinical trials is using patient-derived xenografts together with Baylor College of Medicine and the University of Utah Huntsman Cancer Institute. And we've identified 30 patient-derived xenografts from patients with triple-negative breast cancer, which is the one for which optimization of therapy is much more needed because they're generally referred just for standard chemotherapy. And with these 30 patients, we have them in 2 groups here, 15 that have an average survival of only 1.34 years, and 15 that have an average survival of 7.63 years. And so from these, we'll be growing them in the mice, we'll be isolating circulating tumor cells with the VTX-1 and testing out these procedures for testing the precision medicine applications in live circulating tumor cells that we're currently also undergoing prospective clinical trials here with triple-negative breast cancer patients at the University of Maryland. And interestingly, for these PDX from Baylor and Utah, they already have data available for complete RNA-seq, copy number variation, and mutation. So we can relate that to the phenotypes of the live tumor cells in the tumor site, as well as in the circulating tumor cells. And this is something that we've reported in the last couple of years together with a group of Kostas Konstantopoulos at Johns Hopkins, looking at how live tumor cells migrate through microfluidic devices for breast cancer cell lines, and those with high metastatic potential are better able to migrate through these channels. And then we also connected, so this was just for breast tumor cell lines here in this 2019 Nature Biomedical Engineering paper. But then we had another one in 2020 that used primary tumor cell lines from glioblastoma patients. And there we are actually able to connect the migratory cells, their ability to pass through a narrow channel, and their staining for Ki67 with patient outcome from those glioblastoma cells. And so what we found was the combination of the migratory cells that could go through small channels and Ki-67 staining gave us the best area under the curve for predicting the prognosis of those patients and their clinical outcome. So these are the types of things that with live tumor cells, you can actually measure what the potential outcome is for those patients and also potentially measure drug interventions. And so then just in closing, I mentioned to you before, like rather than trying to find more complex ways to grow the tumor cells, we're trying to see what we can do with those tumor cells right away by isolating the live patient CTCs. And this is where the Vortex VTX-1 really fills a critical gap in our research, was that ability to translate what we can do in live tumor cells in our mouse and zebrafish models to actually begin to do that with patient blood samples. And the seamless integration with the VTX-1 and our TetherChip allows us to rapidly measure these phenotypes in terms of what they indicate in terms of outcome for the patient, as well as their drug response. All right, and so in closing, I'd just like to thank the members of my lab, Vortex Biosciences, for this incredibly productive collaboration using the VTX-1 to isolate live cells from blood samples, as well as our collaborators at University of Patras, here at the School of Medicine, and as well as the bioengineers at both Hopkins and University of Maryland, College Park, and the number of agencies that supported our work. But importantly, the people in the lab who really did all the studies that I told you about today. Kiata is our preclinical specialist. Julia is the one who really refined the TetherChip technology, and everyone else is contributing projects to that. So thank you very much. I'd be happy to take questions. Together with Elodie, it looks like we have quite a few in the questions and answers. So I will give this back now to Chris for moderating the questions.
Stuart, that was fantastic. A really amazing presentation. We've got quite a few questions. So if I just kick off straight away, that would be great. One for you, Stuart. What do you think is the best therapeutic window for targeting CTCs?
Yeah, that's a great question. So I think this comes out of the 2017 Science Translational Medicine paper that I told you about from Albert Einstein, where basically when you give chemotherapy to a patient who has a tumor, it poses the risk of scattering those cells into the bloodstream. So we really see a therapeutic window for one of the immediate therapeutic windows for CTCs, as well as microtentacles, would be at the time that you treat that primary tumor with something like neoadjuvant chemotherapy. You might also want a therapy on board that inhibits the reattachment of the circulating tumor cells so that any CTCs that are scattered by the initial therapy are actually less efficient at reattaching. And so that, I think, would be the short-term window. And then, of course, technologies like the Vortex would allow you to follow that longitudinally in patients over many years as well. But there might be a short-term window where you could be very effective. So, great question.
Super. Thank you, Stuart. A question from Paul Hoffman. Any data of indigen expression or indigenous expression in the tips of the tentacles?
Right, integrins for cell matrix attachment. Yeah, so we have looked at integrins, we've looked at cadherins, and we've looked at galectins on the surface of tentacles, and we have not found anything that specifically concentrates at the tips. So our simplest explanation is the tentacles just non-specifically extend the surface of the cell, and what's already on the surface is going to help those cells attach where they need to go. Now, that might just mean that we haven't found the right receptor that is concentrated at the ends of the tentacles, because there's a perfect superhighway to transport receptors to the ends of the tentacles, which is the microtubules, right? The microtubules transport lots of vesicles, so they could transport specific receptors out to the ends of the tentacles, but we just haven't found anything yet that specifically concentrates there. But we have looked a lot. So, but it's a great question. Thanks.
All right. Last one from Paul. How many CTCs do you need for CDX is in zebrafish?
So we currently inject just a few hundred cells into the zebrafish because we don't have a very large volume that we can inject into them. We're injecting into about 36-hour-old embryos in the zebrafish. And so we have not established CDX, which are like the CTC-derived xenografts, certainly not in the zebrafish. And we haven't done that in mice either. Some people have had much more luck with things like lung cancer cells for establishing CDX than breast cancer cells. We are working with a number of different technologies to try and culture out the breast cancer CTCs. And so even though we're interested in the very rapid analysis, if we can establish a growing culture of the cells, it's of course an advantage as well. And what we want to do is compare the freshly isolated cells with the ones that have been cultured. So we are using methods for that, but not in the zebrafish. The zebrafish is primarily an imaging platform because it's beautifully transparent. You can see the cells and they're circulating together with blood cells and intact blood vessels. And the zebrafish makes all that in 36 hours, which is a lot easier than trying to build that system yourself in vitro.
Excellent. Thank you, Stuart. I'm going to bounce some questions over to Elodie, if that's okay.
Sure.
So Elodie, Amir asks, what additional applications could you find for CTCs by separating them after achieving an automated and repetitive process in the VTX-1 machine?
I guess the question is what applications we can do with the CTCs isolated. So the applications that can be pretty broad. There are a lot of people using especially the CTCs for genomic assays. So DNA mutation analysis with next-gen sequencing or targeted sequencing, or RNA, so transcriptomic assays, either as pooled cells or single cells. Some people are starting to consider methylation markers as well on the CTCs isolated. So that's for nucleic acids, but you can also do proteomic assays, Western blotting, immunofluorescence tagging, functional assays. You can do multiple things, really multiple applications with the CTCs. I understand that was the question.
Okay, thank you. Thank you very much. And another question from Paul Hoffman to Elodie. Any experience in SCLC? And what is the limit?
What's the limit of the size? So we have done mainly, so we have done cell lines of both small cell lung cancer and non-small cell lung cancer cell lines. However, for patient samples, we have done only non-small cell lung cancer for EGFR mutation analysis, for example, or for enumeration. That said, we would love to try small cell lung cancer patient samples if we had the opportunity to do so. And the cell size, so it's difficult to have a very strict cutoff. That's why it's difficult to really know for sure about small cell lung cancer, because it's not only the cell size, but also the deformability of the cells that matters in the separation process. Overall, in general, our cutoff is around 12 micrometers, but that's really more like a Gaussian cutoff. So we know we also capture smaller cells, smaller than 12 micrometer cells. But again, as the deformability matters as well, the best way to know would be to try real patient samples, and we would love to do that.
Excellent, thank you. Another question to you, Elodie. Have you applied this technology to multiple cancer types? If so, which ones? And can this apply to other tissues and any other types of cancer? So
in theory, this can apply to any type of tissue cancer. So I don't count, obviously, here leukemia, lymphoma, and so on. But any type of tissue, we at Vortex specifically, we have done colorectal, breast, prostate, and colorectal, breast, and lung, non-small cell lung cancer. So we have done that internally. We have users that are applying the Vortex technology to pancreatic cancer. And some have also tested ovarian cancer, head and neck. So yes, in theory, I would say any type of tissue cancer, but this obviously has to be tested for any claims, but yes.
Excellent, thank you for that. Back to Stuart. Stuart, why are microtentacles observed in so many different types of cancer?
Oh, thanks. That's a great question. So what we found by looking at the molecular details of the tentacles is that the modifications to microtubules and the roles of intermediate filaments like vimentin mimic what you would find at an epithelial wound edge. So studies have been done over many years. If you scratch an epithelial layer, you'll actually get stabilization of microtubules and upregulation of vimentin at the wound edge. So our interpretation of that is the tumor cell that's free floating in the bloodstream, like a CTC, is encountering a wound in all 3 dimensions. And so that's why it's reaching out with these structures to try and find a partner. And in terms of human cancer, about 90% of human solid tumors are carcinomas, which means they arose from epithelial cell types. And epithelial cells in the body are tasked with barrier function. So when they're separated from each other, they make this aggressive attempt to re-engage each other. And so that may be why we find the tentacles in so many different cancer types. And that sort of persistent wound healing effect is helping them lodge and invade distant tissues. So we think that's why it's a common principle. And so as Elodie had mentioned, the seeing in different cell types, we've also seen, as I presented, tentacles in colon, prostate, lung, and breast cancer. But then we also have collaborators here who have observed them in ovarian cancer as well.
Okay, great answer, thank you. Why do you think different types of chemotherapy can promote metastasis as well, Stuart? Thank you.
Well, so that sort of follows on the previous question, that the chemotherapies, as were shown by Albert Einstein College of Medicine, the increased metastasis was caused by many different therapeutically distinct chemotherapies, so DNA damaging agents or microtubule stabilization agents. And so I think what we need to realize is those chemotherapies are very harsh. And so they're going to impose a wound healing response in the tumor cells that survive, right? So chemotherapy is targeted at destroying and killing the tumor cells. But the question is, the ones that are left behind, the ones that scatter into the bloodstream, has the chemotherapy made them more aggressive in terms of their wound healing responses? And so that's why I think that a number of different chemotherapies could have similar effects.
That's super, thank you very much. Back to Elodie, if that's okay. Elodie, have you tried other types of body fluids other than blood?
Good question. Yes, we have. So we focus mainly on blood because, yeah, that's the most noble fluid, and that gives the most of the content. But we have also tried pleural fluids for lung cancer, peritoneal fluids, several urine samples as well. Yes, so, and also Buffy-Coat samples. So not blood directly, but we can also do Buffy-Coat samples as well. So yes, other fluids, and that's something we would like to pursue at some point as well.
Excellent. And with that in mind, have you tried any other cellular targets other than CTCs? That's
a very good question. Yes, so we tried. So one user came to us and asking about fetal cells and prenatal testing. And so we tried first with trophoblast cell lines at Vortex to make sure that it was working fine in our hands first. And then they tried and it worked. And they actually got a publication out of it that was published end of last year. So that's a good, perfect transition. Yeah, so it's pretty, it's pretty neat. They can isolate the trophoblast, and then they do genomic analysis of the cells isolated to confirm the male fetus from the maternal blood. That's cool stuff.
Excellent. And last question to Elodie on that basis is, how do you compare with ctDNA?
How do we compare with circulating tumor DNA? So a lot of people always believe that these are two competitive approaches to competitive liquid biopsies, and that should be either ctDNA or CTCs. But actually we believe, and others believe as well now, that these are very complementary approaches because they basically provide different content. It should not be one or the other. It should be both together as a complete liquid biopsy approach. With circulating tumor DNA, you have small DNA strands, so you can really look at specific mutations or methylation markers and so on. And it has been well demonstrated for early diagnosis of the cancer or monitoring of recurrence. While CTCs, with CTCs, you have access to the full cells, so to the cellular entity. So you can look at RNA, DNA as well, but longer DNA strands. So RNA, proteins, you can also grow the cells and do functional assays. You can use the CTCs early stage as the circulating tumor DNA but also to look at a response to a given treatment, as Stuart Martin explained today, or to also detect recurrence. So that just to say that these two liquid biopsies are not really competition. They just they're useful at different time points of the cancer care, and that they should be considered together for a better vision of the cancer. But we have done studies specifically where we compared both, for example, for EGFR mutation in non-small cell lung cancer patient cohort. That was done with UCLA, and this is a publication also on our website. And exactly as I said, sometimes there were cases where the mutation was only caught in the circulating tumor DNA, sometimes only caught in the CTCs, and most of the time that was detected in both. That we have done. So circulating tumor DNA and CTCs together from the same tube of blood.
Thank you, Elodie. Great answer. Stuart, back to yourself, if that's okay.
Sure.
A couple of questions. What capabilities of the VTX-1 are most important to you in your studies?
Oh, for us, as I mentioned, it's the fact that you're not subjecting the cells to tremendous physical pressure by trying to wedge them through a pore. It's label-free, it's rapid capture, and it's getting the live cells. And so just to sort of follow on what Elodie was talking about before in terms of comparing CTCs and ctDNA, you know, we can use the live CTCs to judge which therapy might be the best for the individual patient. If you're monitoring patients with ctDNA when you're treating them, you're treating the cancer that's still in the patient. And as we can appreciate from the current studies about chemotherapy scattering CTCs, there is some risk to that, right? Like you're giving the therapy to a patient and measuring their ctDNA. So you're doing that treatment in the patient to test which is the best therapy for them. And that's potentially a little risky. So we're hoping to go beyond that by using the live cells to do that drug testing on the isolated cells. And the Vortex allows us to do that because it rapidly isolates the live cells, right? We couldn't do this for years because so many of the technologies relied on chemical fixation of the CTCs for their isolation, like the cell search does in many cases. And I think, as you saw, the fixation itself destroys the tentacles. So studies that are using a fixation step up front are destroying these structures, which we think are interesting to follow. Yeah.
Excellent. And with that, how do you envisage or envision incorporating CTC analysis with the VTX-1 into patient treatment, for example, taking in the next step?
Right, and that's using the multiple different phenotypes that we can use from small amounts of live cells. And so we've been able to show feasibility for doing that for tentacles, for the stem cell sphere formation, as well as the ability of the cells to cluster. The clustering is a little bit trickier because we need more cells to actually have them cluster together. And so that's why we're working on trying to expand the cells in culture as well for a short term. We're still trying to stay to a minimum amount of time that the cells have been out of the patient to reduce the variables that would change from that patient, right? If you grow the tumor for 6 months or 12 months in a mouse, you may have changed a lot of the characteristics that were important for the original patient. So that's why we like the VTX-1 for its rapid isolation, because those phenotypes aren't going to change nearly as much in 1 hour as they're going to change in 12 months in terms of relating it back to the patient for which therapy they should take. Yeah, thanks for the question.
Thank you, Stuart. Thank you. One last question from Jeffrey Bryant. Jeffrey, I will answer your question by email, but thank you very much for giving it to us directly. I have no more questions at the moment, so I'd like to say thank you to both of our speakers at the moment. To Elodie Sollier, thank you very much indeed, and Professor Stuart Martin. Really appreciate your time. It's been a fantastic webinar, very, very informative, and I'd like to thank you all for attending. Much appreciated.
Thank you very much.
Thank you.
So joining.
Thank you.